Okay, we're good. We're going to get going. All right, everybody. Thanks for bearing with us for the audio-visual difficulties and for braving the weather to come today to Mireia's talk tonight. It's going to be the last talk of Boston Protein Design and Modeling Club of the year. Next year, just to say, next year, it's actually the last talk of the Boston Protein Design and Modeling Club in this room. This room is going to be renovated over the new year and into the following year. So we're going to have to figure out another room. Stay tuned for that. It won't be here though. So enjoy the last time in the Cannon room for pretty much anybody who's going to be in the Cannon room. Let's see. So that's it. And the last thing I want to say is I should thank Twist for donating to our seminar series, helping to sort of support the protein design community in Boston and elsewhere, pizza you're eating and the drinks that you're drinking. You can thank Twist. So I won't belabor the introduction. So Mireia is the head of immunotherapeutics at AI Proteins. And she's going to talk to us today about design of mini proteins that engage NK cells. Mireia came from Germany. She did her PhD in Germany, working on the nuclear core complex and figuring out binders to the core complex through display methods. And then started in Chris Ball's lab at the IPI when they were at when Chris was at the Institute for Protein Innovation. And then when Chris started up AI Proteins, she and a few other old postdocs went with Chris to start up the company. And I think that what you're going to be talking about is going to be work done at AI Proteins. Is that right? Okay. So with that, I'm going to give the floor to Mireia.
Yeah. So hello, everyone. First of all, thanks for making it here in this crazy rainy day. I'm very happy to be giving the last talk of 2024. And yes, today I'm going to talk about the novo-designed miniproteins and why we think that they are great building blocks to engineer immune cell engagers of next generation. Before getting into that, however, I would like to take some time to acknowledge the team. All the work I will show today was actually done at AI Proteins. This is a picture of the team as last summer during our company retreat in Vermont, and I am very grateful to work with all of them. But for the talk today, I want to especially acknowledge Chris Ball, who is the CSO and president of the company, and James Kinali and Xavier Michelet, with whom we've been working close together in this project. So if we want to engineer amazing immune cell engagers, the first question is what an immune cell engager is. And immune cell engagers are multivalent miniproteins in its simplest version of 2 different specificities and 2 valencies. So they can simultaneously bind to a tumor-associated antigen. This would be a receptor that's overexpressed on the tumor cell surface, and to an activating receptor on the immune cell surface. By doing that, immune cell engagers recruit immune cells to the tumor site, and since often they have cytotoxic activities, they release cytotoxic granules that would eventually trigger tumor cell apoptosis and cell death. The first immune cell engager that was approved was the now well-known blinatumomab in 2014. This increased the interest in the field, and between 2022 and 2024, 8 more immune cell engagers have been approved, which is remarkable. The most recent one was earlier this year. It's this tarlatamab. And this was very exciting because it targets small cell lung cancer, which is a solid tumor with very few therapeutic options. But what do we do now? How do we make this next generation of immune cell engagers, and we can get more molecules in the clinic to show safety, efficacy, and be approved for a wider range of cancer indications. We are thinking of different issues that have to be addressed. One of them is to increase the specificity with which we target tumor cells. The issue here is that these tumor-associated antigens that are expressed on the tumor cell surface are also often expressed by healthy cells, which leads to off-tumor on-target toxicities. One way to address it would be to have a higher control over the affinity with which we target them, or even target multiple receptors simultaneously to be able to play with on-cell avidity effects. Another critical issue is how we go after immune cell activation. So we need to decide for each indication which immune cell type we want to engage, which receptor we think will be the most optimal, and maybe it's better to engage two different receptors at the same time, maybe one activating receptor and one co-stimulatory receptor, or even we could think of engaging two different immune cell types at the same time. Another point especially important for immune cell engagers is to overcome the immunosuppressive tumor microenvironment, because if we want these molecules to work, we do need immune cells that are functional at the tumor site. There are different ways of doing that, but we could target these immunosuppressive cells that are always present there and inhibit pro-inflammatory anti-tumor functions, or we could, for example, inhibit checkpoint inhibitory pathways. And then finally, one question that we are very interested about is how we can engineer this immune synapse. So we are thinking about, is there an optimal distance between the two cells or a best geometry of engaging them that would put immune cells in the best position to kill the tumor cells. So what all these aspects have in common is that we probably need multifunctionality, meaning combining different functionalities in the same molecule, and that we also need a better and higher sophisticated control over what our molecules are doing at the cell surface. So far, that's a slide. Okay. So far, all the immune cell engagers that have been engineered are made with antibodies, antibody-derived fragments, or other proteins that can be found in nature. For a Boston architecture analogy, this is the Trinity Church, built in Boston in the 1800s. And humanity also built its first buildings using natural materials, such as stones or clay. However, in order to build more sophisticated buildings, such as the skyscraper that was built more than 100 years later behind the Trinity Church, more sophisticated materials had to be used, such as concrete or steel, so we could have more sophisticated control over their properties. Following this line, what we do at AI Proteins is to design mini-proteins de novo, meaning that we make them completely from scratch using just computational tools, which also means that they do not have any evolutionary history. Therefore, we can engineer them to be wherever we want them to be. So the question here is, which features do we want mini-proteins to have to be excellent building blocks to make immune cell engagers? This structure in the slide is a crystal structure of an actual mini-protein. In this case, a mixed topology with one alpha helix and 3 different beta sheets. And mini-proteins usually have a molecular weight between 3 and 5 kilodaltons and a length between 40 and 60 amino acids. However, despite of its small size, they are distinguished from peptides because they do fold in a very highly stable 3-dimensional structure. This increases a lot their stability, for example, to temperature or to protease cleavage. In addition to this high stability, we can also make binders with fine-tuned affinities. The optimal affinity will depend on the final application, but very often we can make binders with picomolar dissociation constants, and I will show a few examples of this later. In addition to that, we can engineer them to bind to our targets very specifically. And examples of how we can do that is to, for example, avoid having positive charges on their surface, which could recognize cells unspecifically, or avoiding having too many hydrophobic residues at the surface to avoid unspecific protein-protein interactions. The fact that they have such a small size also favors its low immunogenicity, because it's less likely that immunogenic sequences are found. However, we also have tools that I will not go into that today, but we do have tools to predict the immunogenicity of these proteins and engineering it off if necessary. Another advantage is that since they are made only of canonical amino acids, we can express them recombinantly in different systems. It is, for example, very useful to make them in E. coli if we want to make them very quickly and in a high throughput manner. However, we can also express them in yeast or mammalian cells if we want to have them, let's say, endotoxin-free. And then finally, and very important for the story that I will explain today, we realize that miniproteins are highly modular, meaning that we can fuse them together using genetically encoded linkers, and they retain their properties. For a more deeper example into that, here you can see different examples of proteins of different topologies, and now they are all still monovalent. For example, the green one is made only of alpha helices, but the others combine different topologies in different manners. The first thing that we would probably do to make an immune cell engager is fuse together the green miniprotein, and let's say this targets a tumor cell specifically, with the yellow miniprotein, which could target an immune cell receptor. And here we have already a bispecific immune cell engager. This is the modality that most antibodies that are in the clinic still follow, but with miniproteins, we can bring this concept much farther. And we can start asking, what if we want to target two different tumor-associated antigens at the same time? Or what if at the same time we want to, for example, inhibit a checkpoint inhibitory pathway? We can even bring it farther and say, will it be better if we have two copies of this binder to the immune cell receptor? And maybe it's even better if these two copies of the yellow immune cell receptor are actually next to each other. So overall, with miniproteins, we have a huge set of tools that we can use to plug and play and engineer any functionalities that we want at the immune cell surface. To give an example of that, today I will talk about an NK cell engager targeting AML, or acute myeloid leukemia. AML is the most common type of leukemia among the adult population, and historically it has been pretty challenging to develop immunotherapies because the lack of good tumor-associated antigens. Most of the receptors that are expressed by these cells are also shared by healthy progenitor stem cells, which gives place to side effects. And for the non-biologists of the room, just a bit of background of why we chose these targets and cell types. We like NK cells because they are part of the innate immune system and the first line of defense against tumors. It has also been observed that they have an important role in graft versus leukemia. This means that if you engraft immune cells from a healthy donor to an AML patient, it has been observed that NK cells are the ones with a predominant role in killing these tumor cells. In addition, they are believed to have a favorable safety profile in the clinic, and we did choose CD16A as our activating receptor because it's a very well described activating receptor, but it is also an Fc receptor, meaning that if you work with antibodies and they do have a competent Fc domain, they are probably actually engaging NK cells by binding to CD16A. On the tumor cell side, we chose LILRB4 as our target. This is not one of the common targets that are usually used for this indication, but we do like it because it's expressed in some AML subtypes and not at all on healthy progenitors. In addition, LILRB4 is also expressed in immunosuppressive cells that are found at the tumor microenvironment. So if we can, in addition, kill them, it's like killing two birds with one stone. Okay, so how do we do that at AI Proteins? First of all, and as I said before, we design mini-protein scaffolds with different topologies and very high sequence diversity, and we do that completely computationally. At this step, we only care about them being well-folded, stable, and adopting the structure that we want them to adopt. But then we introduce targeted diversity at their surface. By doing that, we can then build phage and his display libraries that we screen using proprietary methods. Our yeast and phage display methods are then coupled to a next generation sequencing NGS pipeline, so we can get an overview of all the sequences that have been enriched. And then we produce what we think are the best hits using our high throughput automated E. coli production platform that can make up to 1,000 different proteins a week. As an example of that, I'm going to talk about CD16A. This is, again, this activating receptor expressed on the NK cell surface. In blue there, you can see the crystal structure of its extracellular domain. And the challenge at this point is that there is a very similar protein called CD16B, which is expressed in neutrophils, but not in NK cells. We did not want to engage neutrophils because they are much more abundant in circulation than NK cells. So they could act as a sink and decrease the efficacy of our mini proteins. And in addition to that, it's not completely understood whether neutrophils actually play a beneficial role at the tumor site. However, the challenge here was that the difference between CD16A and CD16B in the extracellular domain, it's just two different point mutations that you can see in reds in the crystal structure. So it was a perfect test case to investigate the specificity of our mini proteins. So we used this protein, we screened our libraries, and even though, as expected, we did find many binders that cross-reacted with both CD16A and B variants, we also found some proteins that did show some specificity for CD16A. One example of that you can see in the SPR plots, and this is one protein that shows an association and dissociation curves for CD16A, but not binding at all for CD16B. Another sanity check that we usually do at this point is to make sure that our hit binders fold in the way that they are supposed to fold. We do this by circular dichroism or CD, which if you've worked with small proteins, you probably know that's a biophysical technique aimed at measuring the secondary structure of proteins. So in blue here, we can see the alpha fold prediction of this hit binder, and the CD spectra does reflect the trace of what we would expect for a mixed topology containing both alpha helices and beta sheets. In green, we can see this spectra trace at 25 degrees. We see that when we heat up the protein to 95 degrees, the structure is completely lost. But when we cool it down again to 25, it adopts the exact same structure as before, meaning that the protein is able to refold after heating. So we were pretty happy with this protein. It has the specificity that we want. It does seem to fold as expected. The problem is that if you look at the SPR trace, the binding is pretty weak. And if we look at the association, at the dissociation curve, it pretty quickly goes to zero. It was that weak that we could not even fit a reliable dissociation curve, but the affinity is definitely higher than 1 micromolar and too weak for what we need for our applications. So for these cases, we do have an affinity maturation proprietary platform. We call this awesome, and our favorite joke is that awesome is not an actual acronym, so don't try to find words that fit it. It just derives from SSM, single saturation mutagenesis, and we just called it awesome because we think it's awesome. So how does awesome works and how it's different than the traditional methods for directed evolution? We know that directed evolution always starts with an initial sequence and that it introduces mutations with the aim of improving its features. In this case, its affinity. We also know that the whole sequence space cannot be sampled at the wet lab, even for such small proteins as miniproteins. So what most people do is to introduce mutations randomly, and one example of that is error-prone PCR, where an error-prone polymerase introduces random mutations at random positions. The problem of that is that you end up with a lot of sequences that are not functional and are not functional for reasons that could be predicted, such as a bulky residue at the core, which will break the structure, or an exposed cysteine that will give place to the formation of undesired disulfide bonds. It also means that very big libraries need to be built in order to find one improved sequence, and that an optimal sequence might actually never be found. So how is awesome differently? Awesome is a combination of synthetic biology and artificial intelligence to introduce diversity in a targeted way. This means, for example, that all the mutations that are predicted to break the structure would not be included, and that each sequence that's in there has a higher possibility of becoming an actually improved sequence. This allows us to build libraries that are much smaller, and we can quickly, again, screen them by yeast display plus NGS analysis to find a toolbox of binders with a wide range of different affinities, while at the same time producing a huge amount of data that we can use to train future awesome models. So going back to this CD16A binder, again, extremely specific, but too fast payoff for our goals. After undergoing this awesome process, we could find this new binder, which now still binds to the two allelic variants of CD16A, but the payoff is much slower. We are able to fit an accurate kinetics equation, and we got a binder that has single-digit nanomolar affinity, meaning that improved at least 1,000-folds. What is more exciting, however, is that even after this increase in affinity, it does not bind at all to any of the two variants of CD16B. Again, as a sanity check, we ran this protein through the CD spec to confirm the fact that the awesome process did not alter the folding of this protein.
You didn't explain the method, but your drawings made it look like it was a fret pair, and yours, you look for...
The awesome methods? Yeah, sorry.
Is that the principle behind it?
You said fret? Sorry, the question...
I don't know. This picture, to me, you didn't explain it, but if I look at the drawing, I interpret the drawing as like fret, or when it binds, then it turns on a signal or something. I mean, I don't know what this card... You didn't...
Yes, exactly. So the question was about how the awesome method works.
Yeah.
We haven't disclosed it yet publicly, so I cannot talk about the details. It's not based on fret. The picture tries to depict how yeast display works. And the red and the green are just two different fluorophores for how we look at binding and at display of these proteins.
Hello. So the 1000-fold improved binders, were they single point mutants?
No.
Okay.
These binders have 4 mutations as compared to the initial one.
Okay, thank you.
Yeah. So even though the awesome is derived from single-site mutagenesis, we do introduce more than one mutation at once. So we also have the possibility of looking at epistatic interactions. Okay, so moving to the tumor cell side, again, the target that we selected for that purpose was LILRB4 because it's highly expressed in AML and not on the healthy counterparts. LILRB4 is part of the LILRB family, which consists of 5 different proteins with also some structural homology and some sequence identity as well. We went through a very similar process, and we also obtained a binder which is highly specific for LILRB4, the protein that we're interested in targeting. And in this case, it does have a picomolar affinity. Its AlphaFold structure prediction is also a mixed topology, even though it is a little bit different than the one of the binder I showed previously. And the CD-SPECT also confirms this mixed topology, that it unfolds when boiling it to 95 degrees, but it's capable of refolding back to the exact same structure if we cool it down again.
Mireia, I have a hopefully quick question. When you are initially designing the proteins, are you targeting a specific epitope of the target that would distinguish, if you were to accurately bind that epitope, that it would not bind these other proteins that you're showing? Or so I'm just at, like, where does the specificity come from? Is it by design, or is it sort of, hey, look, it's specific, we don't know why, we didn't even target an actual epitope? So can you just say a little bit more about the epitope?
That's a great question. So when we do it with the method that I just talked about, the screening our libraries, and in these 2 examples that I showed, it did happen by luck. Like, we had the ability of sequence all the enriched binders, and a proportion of them happened to be specific. When we matured the CD16I binder, however, we did include negative selections with CD16B to avoid the risk of losing the selectivity during the maturation. However, we also have different methods where we do targeted design from scratch instead of using our libraries, where then we can have a better idea of which epitope we'll go after, and if we want selectivity, we can choose this epitope.
I see. So, so if I'm understanding correctly, it's sort of like you started with, if I go back to your pipeline of computational design, display, et cetera, that you sort of use the computational design to build up as like a starting point for building up a large library that could be a general library for any target, and you sort of screen that against this target.
Yes.
So it's not like you were targeting a specific epitope in this protein. This is sort of a general library, throw at anything, see if something binds, then do affinity maturation.
Exactly.
I see. Okay, thank you.
Yeah. Yeah, we can talk more later, but definitely. Okay, so when we have the binders to all our different components at the specificity and affinity that we want, then it's when it gets fun and we can start engineering them functionally and building multivalents. By doing that, we can start asking questions such as what's the best balancing in which we want to combine them, what the optimal geometry at the synapse will be, and what is the best distance for this synapse. So just as an example, again, we would probably start engineering a bispecific just to prove that this is functional, but then we could, for example, evaluate whether having a second NK cell receptor binder would improve the potency of our molecules by, for example, engaging 2 NK cells simultaneously, or maybe it is better to have these 2 binders close to each other and with a shorter linker, so we can have a higher on-cell ability. To answer all these questions, the kind of functional assays that we do are cytotoxicity assays. And to explain this very quickly, we co-culture tumor cell lines that do express our target of interest, in this case, LILRB4, with primary NK cells isolated from healthy donors. And after some time, we can use flow cytometry to look at how many tumor cells are now dead, and if this correlates with the expression of NK activation markers at the cell surface. So in the legends, the CD16A binder will always be in blue, and the LILRB4 binder will always be in yellow. And the first question that we asked is, if we just build bivalence, as you can see in the gray and black legend, will there be an orientation that will be better than the other? Meaning there will be a binder that will have a preference for having a free termini as compared to the other. And what happens if we build a trivalent? Will this be superior? What we are looking here is at cytotoxicity on the left and NK cell activations, but two very common NK cell activator markers. And the first observation is that there is no difference between the two bivalents. So these two mini-protein binders don't really care in which order they are fused together. But we also see that the trivalent molecule that contains two copies of the CD16A binder, it's superior both in killing potency and in the expression of NK cell activating receptors. After knowing that, the next question.
Just to understand the motivation, why not also try a construct where there's two binding sites for the target, only one binding site?
Okay, so the question is whether we didn't include a construct which has two LILRB4 binders as we did for the construct that has two CD16A binders. I think there are two answers to this question. The first one is that our LILRB4 binder already has an extremely high affinity, which is picomolar as calculated by SPR. So there wasn't really a rationale for thinking that this needed improvement. And second, and this is a bit beyond the scope, I think if we want to bind tumor cells tighter, it is better to use two different tumor cell receptors than trying to make one extremely strong, because this might help switching the equilibrium more towards tumor cells than healthy cells if we play the cards correctly.
In the concentration units, is it concentration of the monomers, or in the case of the dimer, if you have one trimer or one dimer, you count the trimer as twice the concentration as the monomer?
Yeah, so the question is about the concentration units, and if there are three copies, we are accounting for this or not. The concentration is expressed in molar units, meaning that it accounts for the number of molecules, regardless if it's a monovalent, a bivalent, or a trivalent. If we would be expressing this in mass, then yes, this would be a concern.
Wouldn't it be more appropriate to express it in mass? Because it could be that you're it's just one of the two is touching, and then now you have twice as many.
I mean, I think what makes the difference is the number of molecules that you have in the assay.
Number of binding sites.
Yeah.
Can I ask you a question?
It sounds like there's a question from Zoom. Okay, go ahead.
Okay, hopefully you can hear me. Thank you very much. I mean, it sounds fantastic, but quick question I have is on the linkers. Are there slides coming up which speak to the linker choice and linker optimization? Because I would assume that would play a pretty big role on, on the molecule's function. Thank you.
The question is about linkers. Okay, so there's a slide coming up that might partially answer this question. If it doesn't, ask it again.
Okay, thank you.
Sorry, yeah, good question. Do you have the control for when you don't have binders and you just have the CD with like the NK cell and just the
Yeah, so this question is about, I'm just repeating because people don't have mic. This question is about if we have the control of targeting just the LILRB4 binder or just the CD16A. There's also a slide coming up.
I'm still confused about the gray and the black. Are those the same rotationally, or how are they different? Like the yellow, blue versus blue, yellow? How are those?
Yeah, that's a great question. The question is regarding the difference between the gray and the black. The difference is that in the gray, the yellow mini protein has the N-terminus free. In the black, the N-terminus it's fused to the other. And the opposite happens with the blue mini proteins. Okay, good. So we were focusing on this trivalent that has 2 copies of the CD16 binder and 1 of the LILRB4 binder. So we also tried to ask, is it better to keep the tumor binder in the middle, or will it be better if we put it at either termini? Here I'm just showing cytotoxicity data, but it did seem that it's better to keep it in the middle. And just hypothetical, one idea that I do find interesting is that the red construct on the red trace, the one that has the tumor binding moieties in the middle, is the only one that crosses the immune synapse twice. So theoretically, it could help keeping it tighter. And by doing that, assembling more functional immune cell synapse. Of course, this is very hard to prove and just an interpretation. Okay, and finally, and going back to the question of the linkers, when we had set on this trivalent containing 2 copies of the CD16A binder and 1 copy of the LILRB4 binder in the middle, we asked what would be the optimal linker length for this construct. For doing that, we engineered trivalents with linker lengths gradually increasing between 5 and 32 amino acids. And what we saw is that as the linker length was increasing, the potency of our constructs was increasing too. However, we did choose as the best a length of 16 amino acids because the best jump was probably between 12 and 16. And because these linkers are highly repetitive sequences, so by increasing their length, we might also be increasing the other liabilities in our molecules. Okay, so this was our first lead candidate. We called it COC. Again, it has 2 copies of a CD16A binder at either termini, 1 copy of a LILRB4 binder in the middle, and a linker of 16 amino acids in length. Finally, once we had set with this candidate, we just did a few more assays to assess it functionally further. The first thing we wanted to make sure is that in the context of the multivalent, each protein was still recognizing its target by SPR. In blue, we can see the SPR trace of the binding to CD16A. This has increased a little bit in affinity as compared to the monovalent. The reason is probably because now we have 2 copies and there are avidity effects. And the binding to LILRB4 by SPR is exactly the same as the one we had shown before for the monovalent. We also wanted to put this trivalent through the CD spec. Interpreting CD spectra of multivalent miniproteins becomes a bit harder because now we have the contribution of different secondary structures and different topologies, plus the contribution of the disorder linkers. However, it is clear that there's still some protein structure, and what was nice to see and even surprising is that when heating up gradually from 25 to 95 degrees, we did not see any change in structure, which could mean that this trivalent is actually more thermostable than its monovalent parts, which were unfolding at 95 degrees. This is very interesting, and we would not expect to see that, for example, if we would be making immune cell engagers with antibodies, but it supports the point that multivalent miniproteins are highly stable. The next functional question we wanted to ask, and this answers one of the questions before, was regarding specificity. For that, we have, again, our trivalent NK cell engagers, but then we also made versions in which either the CD69 binder or the LILRB4 binder were replaced by the respective scaffolds. The scaffolds would be miniproteins of exactly the same structure, but where the paratop residues are not there, so we do not expect them to bind our targets. The first experiment that we did was against a tumor cell line that does not express LILRB4 because it is not an AML tumor cell line. So here we did not expect any of our constructs to work as we are seeing. However, when we did exactly the same with two different tumor cell lines which do express LILRB4, what we see is that the constructs containing the scaffolds also do not work at all, but our trivalent shows a nice cytotoxicity curve with picomolar EC50s, which proves the specificity of our molecules. This slide corresponds to the same experiment, but instead of looking at tumor cell death, we're looking at NK cell activation, again staining for two very typical NK cell activation markers. And what is nice to see here is that cytotoxicity and NK cell activation correlate very well as we would expect. One more question that we ask is whether our NK cell engager called COC would preserve the serial killing activity of NK cells. In this context, serial killing refers to the ability of an NK cell to kill one tumor cell after the other. Again, in red, we can see our immune cell engager, and the controls are either just tumor cells or tumor cells in the presence of just NK cells. We do that by a live imaging system that can detect individual tumor cells and count them. So what we are seeing on the plot on the right is tumor cell proliferation over time. We see that if we just look at tumor cells, they grow progressively, and actually they double every 24 hours as we would expect. In the presence of NK cells, this growth slows down a little bit, but only in the presence of our engager, all tumor cells are gone after 48 hours. What we do afterwards is to challenge these same NK cells with fresh tumor cells, and we look at the same trends. And again, in rounds 2, 3, and 4, tumor cells with just NK cells keep growing, while the engager is able to reduce tumor cell growth and keep killing them for 4 consecutive rounds, which correspond to 8 days, which I think it's pretty impressive. We also tested our molecule in vivo. To do that, we collaborated with Dr. Todd Feniger at WashU, and we chose an immunodeficient mouse model xenograft. Just to explain a little bit how the experiment works, at day 0, we did engraft this mouse with an AML tumor cell line that does express LILRB4, our target of interest, and a luciferase gene. On day 3, we also engraft NK cells from human healthy donors, and between days 4 and 8 and day N14, we did inject our COC engager daily. Here we look at luminescence images at days 4, 10, and 17. We see that if we have just tumor on its own or tumor plus our engager, but in the absence of NK cells, the tumor grows gradually over the experiment. If we also have NK cells, the growth is slowed down a little bit, but only in the presence of NK cells and our engager, we achieve tumor control. This is just the same data, but plotted. You can see that the black and brown traces correspond to either tumor or tumor plus COC. In blue, we can see how just NK cells achieve some control over tumor cell growth, but in the presence of COC, our engager, is when we achieve the best control over tumor growth. And the last thing we wanted to test is what would happen if we do the same with primary samples from AML donors. This is just to show that if we do that, the experiment gets a bit more complex because of the higher complexity of the samples of these donors. But we did develop a flow cytometry analysis to distinguish the AML cells from the lymphocytes and from our NK cells. Here, the readout is a little bit different. We're looking at CD107, which is a marker that correlates with the granulation and therefore with NK cell killing. And we used it for this experiment because it can be detected very early on. And these AML samples cannot be kept for very long in culture. And what we see again is that for the example of these two patients, in the presence of our COC engager, which would correspond to the green histograms, there is more degranulation as compared to the negative controls. So with that, I showed that we can engineer immune cell engagers using miniproteins, that we can control their valency and their geometry at the immune synapse, and that they work very well in vitro, but also in vivo and with samples from AML patients. But that's just the beginning of everything that we think that miniproteins can do. And I'll just end the talk with the question of, if all this set of tools was available to you, which targets would you like to engage? What do you think would be the optimal affinities, and how would you go after engineering this immune synapse? And with that, I want to thank the team again at AI Proteins, and I'm happy to take more questions.
Okay, questions for Mireia. I have a mic that I guess I could start the first question. Let's see. So is there a critical concentration of how many NK cells you would need to see this effect? Is that going to be an issue with certain patients that just may not have enough NK cells? Or?
Yeah, definitely. So there's always an important parameter that I didn't mention today for this experiment, which is the ET ratio. So how many effector cells are you adding per tumor cell? And you always see that if you titrate this down, the potency of your construct decreases. The availability of NK cells in the patients is definitely a concern. There's also the possibility of engrafting NK cells to this patient together with your protein, and this has been done before.
Thanks.
Hi, great talk. I'm curious, you showed at the start a bunch of different scaffolds for the mini proteins. So you're making a library with all of these scaffolds and you're testing them for binding. But the two you found here, and I'm sure you found many more across other programs, seem to have come from this one scaffold. Is this scaffold sort of uniquely good for making binders? And if so, why do you think so?
They don't actually come from the same scaffold. And actually, even if the topology might seem the same, it's different. So if you go from N to C terminus, one is beta sheet, helix, beta sheet, and the other is beta sheet, helix, beta sheet. So not even the topology is the same. And the sequence is completely unrelated.
Yeah, great talk. I just wanted to ask, you've shown the trivalent structure here. Did you guys go beyond looking at the trivalent structure, and maybe why do you think that, like, if you did go beyond, why did you think like the trivalent structure is the most optimal, or is there something more optimal out there?
Yeah, we have gone much farther. And maybe what I can tell you is I think we've gone up to 12 valencies and it still expresses correctly. And it's superior. The only reason I didn't talk about it today is because that's what we have protected legally so far. But hopefully there will be more to come.
Mouse PK. Do you know how long they stay in there? Like you dosed until day 14, I thought, and then you measured until day 17. But does it go down? How long did it stick around?
So is the question what happens after day 17?
Yeah, like when will they be chewed up?
Yeah, so the issue there and the parameter that we still don't control very well is for how long the NK cells are in the mouse. According to literature, after 2 weeks, they start being eliminated, so we cannot really trust the data after that. Because again, it's an immunosuppressed mouse where we are adding human NK cells.
I also like to talk a lot.
Thanks.
While I'm walking over, sort of related to that question, what is these are still small, even if you fuse them, right? Then you have 3 of them, they're still small. Collectively, like the mass is kind of still less than, I don't know, 15 kilodaltons or something. So I would imagine those would get cleared quite rapidly in just a few hours, right? Just sort of renally. Do you see that, or do you see that they're sticking around?
Yeah, that's also a great question. I think the elimination rate will also depend on how tight you are binding to each cell and for how long. Regardless of that, for this experiment, the reason we dosed every day for 8 days was this, because we were not sure about the half-life and we wanted to be on the same side. However, we're also now developing half-life extension strategies, which I also did not mention today, but we've also seen that they work well.
Does the CD16A domain activate the Fc gamma receptor activation pathways, or is it inert and just kind of acting as an engager?
That's a great question. So what I can say is, according to this data, at least on its own, it doesn't. So on the light gray construct, if you don't have a tumor, if you are not binding a tumor, there is not activation. So at least on its own, it does not activate the same pathways. When it's engaging a tumor cell, then yes, it's the Fc receptor activating pathway.
Since there are no other, there is a hand.
Hi, so maybe I misunderstood about the linker results, but it seemed to be you got better results with the longer linker. And normally, I think the 2 cells interacting would be very close together, the receptor binding to the other protein. And the natural killer, I think, works by secreting something. So I would think that wouldn't closer be better? Perhaps I misunderstood that. I don't know.
Yeah, I think, first of all, the optimal distance there is not very well understood, but there are theories that the optimal distance would be what the natural synapse is. The natural synapse is believed to be around 15 nanometers, which, if just to round it up, let's say one amino acid, it's 0.5 nanometers, and this theory is true, the optimal linker length would be 30. Okay. Of course, this also depends on how proximal to the membrane the epitope is, which we don't know.
So they're not interacting directly, the 2 proteins in the normal. So there's something else in between them in the normal immune synapse.
Yes.
Oh, okay. Okay.
A couple more questions back here.
Yeah, just going off that, do you think the rigidity of the linker would have any effect?
Sorry,
the rigidity. So in the natural synapse, right, it's going to be much more rigid than probably your flexible linker.
Yeah, that's a very good question, and we started thinking a lot about that at the beginning. We still want to keep disordered sequences. The most typical linkers are glycine serine rich, but we usually compare them with linkers that are very rich in proline, which is the most rigid amino acid we can think of. And there are differences, but not very substantial.
Another thing you pointed out was that when you put the 3 together, they were
more thermally stable than alone? What could possibly cause that?
Yeah, that's also a great point. I don't know where I had this data. To be honest, first of all, I don't have an explanation for that. And second, it does not happen all the time. The only thing I can hypothesize around is I don't know if having the termini not free but linked to something else might make it harder for them to unfold.
Do you have data with just each one of those by itself, but with the linker fuse, but not the other?
No.
I think I'm spying my computer, and I think we might have questions from online. Let's see. Yeah, go ahead. Well,
Do you already made the protein bind to the linker, or you just put NK cells and then the linker and hope they bind inside the mouse, or do you make sure that you bind it like in vitro and then you just put it in the mouse?
Okay, is the question if we premix the NK cell? Okay. So wait. For the first day, yes, because they are administered at the same time. But for the rest of the days, and we're administering every day between 4 and 8 and day and 14, this is just the protein alone, because the NK cells are already there, already on day 3.
okay, I've got a couple of questions that I'll read from the chat. Richard Phillips says, great talk. Number 1, how are you delivering the binder? For example, is it expressed from the NK cell? And then number 2, have you tested a similar strategy as a CD16A fusion protein? Do you think bispecific has advantage over such a fusion?
Okay, the first question is whether the NK cell is secreting the engager, right?
Yeah, is it expressed from the NK cell?
Yeah, not in the experiments we've done so far. However, that's a great idea. However, it would involve engineering the NK cell, and it complicates a little bit the manufacturing.
Yeah, and the second one was, have you tested a similar strategy as CD16A fusion protein? Do you think a bispecific has an advantage?
I don't understand.
I don't understand the question either. I'm going to go to the next one. Min Chen asks, have you tried linking a weaker binder for LILRB4 with your CD16A binder instead of using the tightest one? If the engager sticks to tumor cells too tightly, would it inhibit the NK cells from detaching from a tumor cell after it is being killed?
Yeah, that's a great question, and it was the whole rationale behind looking at serial killing. So after seeing our serial killing, we think that's not a limitation. And we have not tried this with the LILRB4 binder, but with other tumor-associated antigen binders, we consistently see that increasing the affinity increases the potency of the construct.
Okay, I think there's 2 more questions. Well, another question from Min Chen. Would using the engager prolong or shorten the lifetime of the adopted NK cells?
If using it for long.
How does it affect the lifetime of the NK cells? I guess using this engager fusion protein.
Oh, if they expand after getting activated and so on. We have not looked at that.
Okay, last question from Needy Jane. Which host cells have you used to express in, and what was the mode of purification?
Did you say CHO cells?
Which host cells did you use to express?
Yeah. So for the high throughput expression and most of our experiments, we use E. coli expression because it's just the most convenient and fastest. And we purify them by affinity chromatography. For the in vivo experiment, in this case, where we needed a sample completely free of endotoxins, we are now using either yeast or mammalian cells. The purification is also affinity chromatography.
Yeah. Okay. If there are no further questions for Mireia, let's thank her again. And that's the last talk of the year. Okay, so stick around, chat with Mireia, eat some pizza, drink some beer, and then we'll see you in the new year.